Biotechnology. Figure 10.1. A thermal cycler, such as the one shown here, is a basic tool used to study DNA in a process called the polymerase chain reaction, or PCR. The polymerase enzyme, most often used with PCR, comes from a strain of bacteria that lives in the hot springs of Yellowstone National Park. Chapter outline. Section 10.1, Cloning and Genetic Engineering. Section 10.2, Biotechnology in Medicine and Agriculture. Section 10.3, Genomics and Proteomics. Introduction. The latter half of the 20th century began with the discovery of the structure of DNA, then progressed to the development of the basic tools used to study and manipulate DNA. These advances, as well as advances in our understanding of and ability to manipulate cells, have led some to refer to the 21st century as the biotechnology century. The rate of discovery and of the development of new applications in medicine, agriculture, and energy is expected to accelerate, bringing huge benefits to humankind and perhaps also significant risks. Many of these developments are expected to raise significant ethical and social questions that human societies have not yet had to consider. Welcome back to Concepts of Biology. We'll continue to read Chapter 10, Biotechnology, Section 10.1, Cloning and Genetic Engineering. By the end of this video, you will be able to explain the basic techniques used to manipulate genetic material and explain molecular and reproductive cloning. Biotechnology is the use of artificial methods to modify the genetic material of living organisms or cells to produce novel compounds or to perform new functions. Biotechnology has been used for improving livestock and crops since the beginning of agriculture through selective breeding. Since the discovery of the structure of DNA in 1953, and particularly since the development of tools and methods to manipulate DNA in the 1970s, biotechnology has become synonymous with the manipulation of organisms' DNA at the molecular level. The primary applications of this technology are in medicine for the production of vaccines and antibiotics, and in agriculture for the genetic modification of crops. Biotechnology also has many industrial applications, such as fermentation, the treatment of oil spills, and the production of biofuels, as well as many household applications, such as the use of enzymes in laundry detergent. Manipulating genetic material. To accomplish the applications described above, biotechnologists must be able to extract, manipulate, and analyze nucleic acids. Review of nucleic acid structure. To understand the basic techniques used to work with nucleic acids, remember that nucleic acids are macromolecules made of nucleotides, a sugar, a phosphate, and a nitrogenous base. The phosphate groups on these molecules each have a net negative charge. An entire set of DNA molecules in the nucleus of eukaryotic organisms is called the genome. DNA has two complementary strands linked by hydrogen bonds between the paired bases. Unlike DNA in eukaryotic cells, RNA molecules leave the nucleus. Messenger RNA, or mRNA, is analyzed most frequently because it represents the protein-coding genes that are being expressed in the cell. Isolation of nucleic acids. To study or manipulate nucleic acids, the DNA must first be extracted from cells. Various techniques are used to extract different types of DNA. Most nucleic acid extraction techniques involve steps to break open the cell and then the use of enzymatic reactions to destroy all undesired macromolecules. Cells are broken open using a detergent solution containing buffering compounds. To prevent degradation and contamination, macromolecules such as proteins and RNA are inactivated using enzymes. The DNA is then brought out of solution using alcohol. The resulting DNA, because it is made up of long polymers, forms a gelatinous mass. Figure 10.2. This diagram shows the basic method used for the extraction of DNA.
All right, let's get back to reading.
RNA is studied to understand gene expression patterns in cells. RNA is naturally very unstable because enzymes that break down RNA are commonly present in nature. Some are even secreted by our own skin and very difficult to inactivate. Similar to DNA extraction, RNA extraction involves the use of various buffers and enzymes to inactivate other macromolecules and preserve only the RNA. Gel electrophoresis. Because nucleic acids are negatively charged ions at neutral or alkaline pH in an aqueous environment, they can be moved by an electric field. Gel electrophoresis is a technique used to separate charged molecules on the basis of size and charge. The nucleic acids can be separated as whole chromosomes or as fragments. The nucleic acids are loaded into a slot at one end of a gel matrix, an electric current is applied, and negatively charged molecules are pulled toward the opposite end of the gel, the end with the positive electrode. Smaller molecules move through the pores in the gel faster than larger molecules. This difference in the rate of migration separates the fragments on the basis of size. The nucleic acids in a gel matrix are invisible until they are stained with a compound that allows them to be seen, such as a dye. Distinct fragments of nucleic acids appear as bands at specific distances from the top of the gel, the negative electrode end, that are based on their size. A mixture of many fragments of varying sizes appear as a long smear, whereas uncut genomic DNA is usually too large to run through the gel and forms a single large band at the top of the gel. Figure 10.3. Shown are DNA fragments from 6 samples run on a gel, stained with a fluorescent dye and viewed under UV or ultraviolet light. Polymerase chain reaction. DNA analysis often requires focusing on one or more specific regions of the genome. It also frequently involves situations in which only one or a few copies of a DNA molecule are available for further analysis. These amounts are insufficient for most procedures such as gel electrophoresis. Polymerase chain reaction, or PCR, is a technique used to rapidly increase the number of copies of specific regions of DNA for further analyses. PCR uses a special form of DNA polymerase, the enzyme that replicates DNA, and other short nucleotide sequences called primers that base pair to a specific portion of the DNA being replicated. PCR is used for many purposes in laboratories. These include 1. The identification of the owner of a DNA sample left at a crime scene. 2. Paternity analysis. 3. The comparison of small amounts of ancient DNA with modern organisms. And 4. Determining the sequence of nucleotides in a specific region. Figure 10.4. Polymerase chain reaction, or PCR, is used to produce many copies of a specific sequence of DNA using a special form of DNA polymerase. In our next video, we'll read Cloning and Genetic Engineering.